Welcome back, everyone. So we move to the next session of our workshop, which is Crafting a Career in Clinical Research. And Professor Vikram Mathews from CMC Vellore has been very kind to join us today. Thank you, Vikram, for being here today to share your journey with the participants of this workshop. Vikram is a physician scientist and he is involved in research and management of leukemia patients for over last 20 years. Vikram completed his MBBS and MD, followed by a special training in clinical hematology from the Tamil Nadu Dr. MGR Medical University. He has then pursued a 3-year postdoc training in Washington University, where he also received a bone marrow transplant fellowship. He will be discussing more about his research and his career journey during his talk. Apart from the various other prestigious fellowships, Vikram has also been a recipient of the India Alliance Senior Fellowship twice. Yes, in 2012 and 2019. He is on the editorial board of Indian Journal of Hematology and Transfusion. He is on the review panel of various eminent journals. So without any other delay, I would like to welcome Vikram to steer this session. Over to you, Vikram.
Thank you, Yukti. I'm just going to try and share my slides, and I'll start straight after that.
So there are three, if you hover the cursor, there should be three buttons, control buttons on your screen.
Okay.
One is for the mic, one is for your camera, and the other one is for your...
That.
Yeah.
Okay, can you see the slides? Can you see the slides?
Yes, we can see them now.
Okay.
Great.
So,
yes.
Okay, can I go ahead? Can you hear me clearly?
Yes, absolutely.
Okay, good afternoon, everybody. I'm sure you've had a busy morning listening to fairly intense talks. The next 25 minutes, I'm just going to walk you through my career. So it's more like a little story, and maybe we can have 5-10 minutes in the end to discuss. So I'm going to walk you through basically my career. And if you read the title, it looks like I had complete control over all of this. And as we all know, a lot of things sometimes come, and it's a question of, reaching out and grabbing opportunities. But also, we are fortunate to some extent to be born in the right place, to have the right resources and opportunities. So I mean, we take that for granted, and I recognize that we really shouldn't be taking all that for granted. But anyway, if I look back at my life, going back to school, this is what I really wanted to do all my life. I wanted to be a hockey player. And if you can see my arrow, that's me over there. I was the hockey goalkeeper for St. Germans. And this was a phenomenal school in terms of, they were more concentrated on sports than on studies. And my first mentor really was a person called Father Mervyn Coelho, who for some reason felt that, I had a great future in hockey, and he really took me under his wings. This group of people were an amazing group of people. Actually, many of them went on to do very well. And this is us almost 30 years later. The person you're seeing next to me over here is Jude Felix, who went on actually to captain the Indian hockey team at the Olympics. I still do play hockey, and it's still a passion. And just to say that even with science, it's always good to have sports and other activities in your life to keep a balance and focus, and that's very important. But at 12th standard, I had to make a choice. I had to decide: am I going to stay in hockey, or am I going to go for medicine? My passion always had been to do medicine, and I thought at that time I had to move on from hockey and actually do medicine. So I went to MBBS in Christian Medical College, Vellore, a fairly routine training, and I finished my period over there, and then I had 2 years of rural service. I just want to mention that, in those days when people used to say this 2 years rural service, they used to look at it as such a burden. You have to go and spend 2 years, you lost time in your career. I can't look back at my life and imagine two more spectacular years in terms of what I actually learned. I spent a year in Chikbalapur, a really small rural hospital in Karnataka, and the second one in a slightly bigger hospital in Mysore called Hallsberg. It's an incredible periods where you actually get to see real world challenges, learning to adapt, using your skills, and in a real practical sense, picking up management and responsibility. So there were many aspects to this, I think, that those who are in this audience who have done medicine may relate to, but those who have not done medicine may also want to look at, actually going out and translating a lot of the theory and learning on the ground from practice. It really does open out your eyes to the opportunities that are there and also the real world challenges that really goes to kind of shape you as a person and your career to some extent. At the end of 2 years, I came back to CMC and I joined MD General Medicine, and subsequently I joined the Department of Hematology, where again I was fortunate. This is what I keep saying. Sometimes these things come across your life, and I had the fortune to work with a pioneer in the field of hematology called Dr. Maman Chandi, and I went on to do what's called the super specialty in hematology. And at the end of that period, my institution allowed me to go for 3 years to Washington University in St. Louis, where I spent some time. And I spent some time with some very incredible people over there. And I came back to CMC, and I've been in CMC since the time that I've come back. While in the US, I met two of my real mentors in science. One was John DePercio, and the other is Timothy Grobert. What I learned from them, what I learned from going to the US, actually, I was already finished most of my hematology training, but I learned certain simple things, how to ask a question. You'll be surprised how important that is. They also taught me, you think that going to the US, it's all because they have all the resources. Not really true. You'll be surprised that we have quite a bit of resource over here. But what I learned was how you persevere on a question and how you continue to work at it, ask the right questions, and how to move in science. So I really think it's very important to have good mentors. And I'm not talking about mentors who are big shots in terms of a lot of publications. I'm talking about people who teach you how to ask the right questions, essentially good human beings. You must also remember that in life, you are certainly going to come across people who are not exactly interested in your career, if not obviously inimical to your career. And you also need to learn how to handle seniors who are like that. And you hope that you will have, be able to find the right mentors in your life. I think that's important, and we should be grateful for them. Moving on, when I came back to India and in my training, I recognized very early that leukemia in India was a completely different ballgame to what you read in textbooks. And this is going to be relevant for you whether you do basic science or public health research or any other form of research. The challenges, the real problems that we face working in our environment is very different from what you may read in any literature or any textbook. And you have to learn to adapt, and you need to answer questions that are regionally relevant, especially in the area of medical research. There's no point talking about this fantastic drug which costs millions of dollars, which maybe Ambani can afford and nobody else can afford. It's got very little relevance to our country. And so we must constantly be grounded in that reality, which is why I really look back at my 2 years of rural service as something that really opened my eyes to what is really relevant for our country. For those who are interested in this topic more, I urge you to go and read this article in the British Journal of Hematology. This is from our group, where we discuss the details of the challenges in treating leukemia in India. Many of these data-intense slides I will not be going through in detail. It's just to give a sense of flow and the perspective of how my work has moved forward. And the challenges with treating leukemia is not just the cost of the drugs. It's the extreme difficulty in accessing therapy. It's the cost of managing the side effects of the chemotherapy. And those are actually the challenges. If we had very specific therapies which cause very little off-target side effects, the cost of managing these diseases would be a lot easier. I mean, all of you all have seen patients with chemotherapy with no hair, lots of nausea, vomiting, severe weight loss. At least you've heard about it. And if you really have good targeted therapy, you really don't need to have those effects. Those are all what we call off-target side effects. And that's something that we need to really work at. And there was a disease called acute promyelocytic leukemia, which I was particularly interested in. This was a subtype of acute myeloid leukemia, which was very rapidly fatal. And as for any other leukemia, only a small fraction of patients could afford conventional therapy because of the extreme complications of bleeding that is seen in this particular type of leukemia. What was fascinating for us is that there were patients who received standard of care, and standard of care, even way back in the early 90s, could achieve 80% cure, but only 10% of our population could afford this treatment. What was very interesting for us is we had patients, when I was doing my training in hematology, who would relapse after these standard therapies. They would go away on what we call palliative care, and then they would come back 6 months later absolutely well. And we found that many of these patients were going to a place in Dehradun to a guy called Balendra Vaidyo, who was an Ayurvedic physician, who was treating them with a substance which we realized was very rich in arsenic. So it's important to keep your eyes open, and when you see unusual events, to try and follow up on those leads. At the same time, there was preliminary data from China saying that arsenic trioxide may work in APL. That really triggered our interest, and we actually looked at arsenic in much, much more detail. And it's actually a very colorful history. It's a drug that's been in the pharmacopoeia for the longest period of time, more than 2,000 years. So as long as the pharmacopoeia existed, arsenic has existed. And it was actually gained a lot of interest in the early 1900s because as the first drug that could actually treat syphilis. It's a non-specific cellular cytotoxin. So if you give enough of it, of course, you can kill anybody. You can kill a human being, forget the cancer cell. And it's believed to have been used as a poison to even kill Napoleon. And that's what the story goes. Now, it's more specific for acute promyelocytic leukemia. So at a very low dose, so what we call pharmacological dose, where there's very little off-target side effects, none of the other normal cells are affected, it seems to degrade the particular oncoprotein called TML-RAR that is very unique to arsenic trioxide. So this is already known. And as a student, I got a grant. Okay, it doesn't sound fantastic. It was about 20,000 rupees. But believe me, even in those days, it was a bit of a joke. But we actually managed to manufacture arsenic trioxide in our institution. This is way back in 1995, 97, for a cost of rupees 1850 per vial of 10 milligrams, which is one dose. Any guesses for what is the cost of 10 milligrams of arsenic trioxide in the market today in the West, not in India? It costs about $400 per 10 milligrams. This also brings another whole topic of big pharma and medicine into the conversation, which we can't go into today. Arsenic trioxide, we had to, after initially manufacturing, we had to move it to industry for various legal issues. Costs about little more than $5 or about 400 rupees per 10 milligram dose. So paradoxically, if you want to use arsenic trioxide in India, it's the cheapest therapy to use in terms of leukemia. If you were to use arsenic trioxide in APML in the West, it's the most expensive therapy. That really is actually a tragedy. And this is Sister Theresia Ba, who at that time was working in the pharmacy and actually used to make this arsenic trioxide. It's not that difficult in terms of a formulation preparation, but we did all the background tests, including the accelerated degradation, animal toxicity, endotoxin assays, etc. And this is the first patient with his permission that I'm showing you, who we treated in 1998 with a single agent arsenic trioxide protocol, which is extremely low cost. And it was very effective, especially in what we call low-risk patients. It's not possible for me to get into the details of this protocol and how it evolved, but it gained a lot of attention in the world and it brought arsenic trioxide to the forefront of acute promyelocytic leukemia. Today, arsenic trioxide is part of the frontline therapy in APL all over the world, in ATRA, and a combination of a drug called all-trans retinoic acid and ATO. When we started in 1997, there was a lot of criticism about, yes, here you have a disease with an 80% chance of cure. People are using the single-agent arsenic trioxide therapy. So you will face a lot of criticism when you do certain things, you must be very clear about what you're trying to do, and your conscience must be absolutely clear that you're not doing this for only personal gain and that there is greater relevance to this. With single agent arsenic trioxide, not the 10%, we could treat practically every single patient who came to our center. And numerous publications followed on this, which elaborated on our experience, both the long and short-term side effect. It was very well tolerated. There were no second malignancies. There was a lot of concern about hepatotoxicity, cardiac toxicity. At the doses that we used this drug, we could demonstrate that none of this was true. Fertility was preserved. So after the initial 2-3 weeks of intensive therapy, because more related to the disease, the entire treatment was outpatient. There was no off-target side effects. So the cost of therapy really crashed. And this is a recent publication looking at the cost of treating APL with generic arsenic trioxide in India compared to what would have been the standard of care. I'm not going to go into the details, but basically to say that the cost is almost 1/4 to 1/5 of conventional therapy. And it's at a cost which is still not easy in terms of the supportive care, but most patients can afford, and those who cannot afford, we can raise the resources in a non-profit organization as the one that I work in. So we treat 100% of our patients at this point in time. The next question that I asked was, unfortunately, in spite of arsenic trioxide, a proportion of patients relapse. And we wanted to ask the question, what is it that makes some people relapse in acute promyelocytic leukemia in relationship to arsenic trioxide? So the conventional thought process as far as leukemia relapses, one is like clonal evolution. So imagine clonal evolution more like Darwinian kind of a theory. Randomly mutations develop resistance to a drug, and therefore that particular clone will survive. And there's another concept called leukemic stem cells. These are cells that lie very quiescent and are not affected by drugs or chemotherapeutic drugs or maybe arsenic trioxide. Through a series of experiments, which I'm not going to go, we demonstrated that neither of these mechanisms are relevant in the context of acute promyelocytic leukemia. And this is work done primarily by my student Errol, who is now a staff scientist in Washington University in St. Louis. And we also collaborated. It's very important in science to collaborate with people. We collaborated with the National University of Singapore. This is a study which looked at the comprehensive mutation profile of newly diagnosed and relapsed patients, combined with this other mechanisms, other data in comparison newly diagnosed and relapsed patients. We proposed, based on the data that we had, that this was not clonal evolution, nor was it a leukemic stem cell population, but in fact, it was a epigenetically modulated upregulation of microenvironmental drug resistance. In short, basically what it means is, if I take a cancer cell and I put it in the peripheral blood or outside the bone marrow, it has a certain amount of viability or susceptibility to a drug. But when I put it into the microenvironment, the same cell in contact with a niche of microenvironment cells, which are the stromal cells give it a signal to survive this chemotherapy drugs. And the mechanism for that is complex and was addressed in a series of papers that came out from our group, again collaborating with people from Washington, from France. We developed the 3D models. Again, I'm just showing this to show that collaborations are so important in taking science forward. No one person can take forward everything, and it's ridiculous to hold on to everything, in the fear that somebody's going to scoop you. I think you need to collaborate with like-minded people, and these collaborations are often very helpful. But we essentially worked out that this microenvironment resistance was primarily mediated by upregulation of the NF-kappa B pathway, at least in the context of APL. And we could also demonstrate work done by my student Sarnan, who is now doing his postdoc in France. We further integrated it and looked at pathways. Again, here we worked with Neha Vyas and Dasrati. These are two Wellcome DBT fellows in different institutions, again illustrating collaborative work, in this case within the Wellcome DBT network, which was very useful. But the bottom line was we were able to translate all this work into repurposing a drug that had never been before used to treat APML, which is a proteasome inhibitor, which could overcome this microenvironment-mediated resistance We demonstrated the mechanism of synergy between these agents, and we also demonstrated an alternate pathway of degradation of the PML-RAR because botusome is a proteasome inhibitor, and the question was, if you combine ATO with botusome, how is PML-RAR being degraded? And we were able to demonstrate an alternative pathway of PML-RAR degradation, which was essential for actually clearing this leukemia. Validated it in a mouse model, and further went on to do a phase 2 clinical trial with this combination in patients with relapsed APL. All this work is available online. And we were able to show the study is not powered to address efficacy. It was basically a phase 2 study looking at safety, but we were fairly confident that this combination was safe. In the small set of number, we could see a signal that suggested what is showing in red is the combination with botusome shows a survival advantage. There's a signal compared to our historical controls, but larger studies and data is required before that can be finalized. So in the context of APML, through all the work that we have done, we could show that generic arsenic trioxide can really reduce the dose, not just because the cost of drug was low, because the off-target side effects were very minimal with this therapy. We have rational and evidence-based data how to stratify patients for this therapy. That's data that I have not shown you, which is based on monitoring the measurable residual disease in patients post the initial treatment, deciding on the amount of intensity that we need to use for them. And this is regionally relevant, data-driven strategies that are relevant for our environment. So for those of you who read, I seriously encourage you to read this book. It's a book called The Emperor of Malds by, the name is skipping my mind, Mukherjee. It's an excellent book. It talks about ALL, acute lymphoblastic leukemia, Siddharth Mukherjee, and the evolution of how treatment of acute lymphoblastic leukemia evolved from the days of a single agent drug to multiple drugs being combined. And today we have almost 80-90% cure in AML. What you see is a list of new drugs that are coming out in AML. So I'm fairly confident in acute myeloid leukemia, which is the rest of AML. Acute promyelocytic leukemia is only a subset of acute myeloid leukemia. The challenges are much more significant. But optimizing these drugs, and in addition, in repositioning and repurposing existing molecules that are already available there, I think in acute myeloid leukemia, we can repeat the story with acute promyelocytic leukemia. And that is what my group is working at. So acute promyelocytic leukemia is a subset of AML where we have very effective therapy. But in AML, which is 80% of the rest of the acute myeloid leukemias, the non-APL AML, the challenges remain in treating these patients. And we are hoping to develop the arsenic trioxide equivalent in the management of acute myeloid leukemia. It's important, again, to collaborate, and I encourage you to visit this website, hemecancer.org. It's free for anybody to register. I'm one of the founding members. Our vision is to work with like-minded people, to be a multi-center collaborative academic organization to improve knowledge, standardize cost-effective treatment strategies, and promote regionally relevant research in hematological cancers. So there are multiple centers and more centers coming along to join with this group. It's primarily clinical-based data sets right now, but we want to involve clinician scientists and also basic scientists to work with this group as we move and try to improve the landscape of leukemia diagnosis and treatment in India. These are some faces to the data of patients treated over the years. Always very grateful to the patients who always continue to place their trust in us. And of course, all this research work is not possible without a bunch of fantastic students that I've had over the years. These are the present bunch and who have come and gone through the lab. And it's wonderful to interact with students, and it's been a joy working with them. Always grateful to them. They do all the hard work with regards to generating all this data. I cannot thank enough my funding agencies, Wellcome DPT. I mean, there's a lot that I missed, but there are periods in life where funding was very, very difficult to get, and especially for a physician scientist, funding agencies either fund basic science or they fund pure clinical trials, and it was very difficult to get the mix, and it came at the right time in my life. Again, highlighting that sometimes in life these opportunities come along, and I guess you just have to grab them. All the students in my department and my colleagues in my department, my collaborators, Dr. Christine Shomi, Dr. Roseanne, Dr. Sanjeev Krishna, Neha Vyas, and Roseanne Padua. I also want to point out for youngsters out there that as you go forward and you apply for grants, you're bound to face rejection, and if not in the majority of cases, as is my experience. So it's important to remember that only 20% of grants actually get approved in most agencies. And when a grant is rejected, it has nothing to do necessarily with the quality of your proposal. It just may be that the grantees were looking for a different kind of studies or a different set of targets in terms of what they wanted to fund. It's also important to remember that funding agencies are looking for you. They want people to take their grants and provide good work, because at the end of the day, they also have to have an output and justify their existence. So when you do get rejections, don't get depressed. This is one way of dealing with it. The harder I try, the worse it gets, and get into a vicious cycle. The other extreme is, boy, everyone is stupid except me. Okay, that's also not a good approach. The balance is somewhere in between, I guess. It's time to review and reassess for sure, but always move on. Sometimes, funding reminds me of this joke. It says, I asked Santa for a research grant, and the reply he got was, you still believe in research grants? It's not so much about the Santa. But whatever happens, have fun along the way. This is me and my son at the Coldplay concert in 2016 in Mumbai. Can you imagine us sitting or hanging around in crowds like this? Great moment. And this last picture is at about 12 midnight with my son. Thank you for your attention. I'm happy to take any questions that you may have.
That was an amazing session, and I really believe that you have been able to inspire and motivate a lot of researchers, young researchers here today. And thank you for highlighting the importance of collaborations in research, and also the importance of having fun in life, and having a balance of work and fun in life. So we have a lot of questions coming our way, and I will just pick some for you. Yes, so how feasible is it for physicians to conduct both basic lab-based and translational research in India? Are such multi-dimensional opportunities available in India, especially in upcoming fields such as regenerative and genomic medicine?
They certainly are. You obviously have to be in the right institution to start with. So once you decide that you want to go into basic science, you need to identify the right institutions, and you need to pursue that right very early that you want to go into that. I mean, it's difficult to walk into a private setup and say you want to do this kind of a research. I mean, it doesn't exist. So it's important to identify where you want to do it, but there are certainly opportunities. I would go to the extent of saying it's not because of lack of opportunities all the time. It's because there are very few physicians who really want to put the time and energy into doing this, because the problem is multi-levels, but it also starts from medical school, where, the whole training, unfortunately, is getting worse these days. It's just theory. There's very little practice. There's very little incentive for being creative or original. The entire incentive and reward system is based on mugging up things and passing entrance exams. And so you have a whole lot of very mediocre people coming out who are probably, intelligent in a certain dimension, but very little creativity. So that's not something that the students here can address. But if you are interested, I would suggest that you persevere. If you're a student, if you're a medical student, find the right center, find the right places that do research. They are there. There are many centers in the country that are doing excellent work, and be ready for a little long haul.
Thank you. We were scheduled to actually close the session at 1:30, but can I ask your permission for a few more minutes so that we can take up a few more questions?
Yeah, it's absolutely fine with me, Yukti. I apologize if I went on longer than.
No, that was absolutely fine. Yeah. So, how important is mentoring for young researchers? How do you find the right mentor in your career?
That's a tough one, Yukti. To be honest with you, a little bit of luck is important. I think it's absolutely critical to find that right mentor. First thing is you need to find mentors who are people who are content and happy themselves. Otherwise, it doesn't work, and you should have a good chemistry with the mentor. I think that's important. That's one side of it. The other side of it, from the student's perspective, you need to be willing to work hard. You can't have only mentoring and saying, mentor is not good. You need to be really willing to work hard. And so it's a two-way situation, but I think mentoring is absolutely critical. It's very difficult in science to just, be placed in a place and then suddenly start performing.
Yes, we absolutely agree with you on that. Here's another one. What would you tell a medical student who is confused between pursuing clinical practice and research? What kind of mindset does one require to be a physician scientist?
I think the step one is you need to step back a little bit and make an honest insight and assessment of what you really want to be. Sometimes it's very glamorous you want to be a certain thing, but then you realize that, listen, frankly, I want to make money and lead a fairly happy life, and that's honest. There's no problem with that. Then choose that path, But if you at the end of this introspection, if you come to the conclusion that you really want to do research and medicine, which is not easy because you will give up a lot of other things in terms of lucrative opportunities, etc. You must stick to that. Whatever you decide, you must be willing to stick to that for the long haul. There's nothing that you can do in 1 or 2 years and say, this is it.
So this is more of a, life skill question here. Please give us some tips on how to deal with malicious people we meet during our research career.
Yeah, I've had my share of them. So I think it's very important not to react. You need to control your emotions. You need to be honest, hardworking, and not necessarily reciprocate whatever is being thrown at you. And I think life is too short. Try to work with people you like and get along with, and do your best, if possible, to avoid people you don't get along with. That's the simple answer.
Thank you. Yeah. So most of
I may just go back to that once, Yukti. So sorry.
Yes.
Because I think the danger of reacting too much is you get drowned in your own bitterness and negativeness, and you actually kills your own work output and creativity. So it's just not worth letting people control you or events to control you. So sorry.
The next one is most of the researchers, especially PhD students, face a lot of mental trauma because of workload, poor outcome, which leads to imbalance both in professional and personal life. This leads to a lot of mental trauma. Can you give some guidance in this regard?
Yeah. I think a lot of this starts very early. Unfortunately, in India, people are not counseled adequately before they take a certain pathway. They often take a pathway. Way back when I finished, there were only two things you could do. You could only do engineering or medicine. There was nothing else. And so if you try, it's basically trying to take a square and trying to plug it into a circle, So if you're not fitting into a certain, and you're in the wrong fit in a particular location, it's going to be hard. It's always going to be hard. So if it's possible, it's good to take and say, is this really what I want to do for the rest of my life? I think the priority really is to be happy. Maybe I'm a better person. I'm a people person. Maybe I should be doing something like that. Maybe I've got better management skills. I think there are plenty of opportunities out there to move away, even within 1 year or 2 years, if you really don't see that this is what you want to do for the rest of your life. I would advise my children that, to be honest with you, because at the end of the day, I think it's better to be happy.
Okay, someone asks, how does the typical life of a MD PhD look like who is more inclined towards research?
Unfortunately, the whole MD PhD stream does not actually exist in an active form in our country. Different people have tried it. It has not really clicked as it has in the United States. The problem is with medical schools to a large extent. They are just so focused on getting and churning out medical programs, whether it's the private sector or the government sector. The whole incentive to research is not there. And that is something that I think the country has to fundamentally address, you know. And the present system of selection of students doesn't help the cause either. It's a reward system which is just based on a certain type of a personality and outputs are going to do well. Unfortunately, we are seeing it, you know, they are not necessarily they may be smart on paper, they are not really the creative people who come out in the system. And the entire mechanism of picking medical students and the super specialists and the postgraduates is going in that route. It's a little worrying times, really, to be honest with you.
We have a question related to your research that you shared with us today. Does arsenic trioxide have any side effects in patients with leukemia with compromised immune system?
Not really. It does have certain side effects. It can have, so you must remember, the guy called Paracelsus in the mid-15th century who said the statement, I think it's very relevant because as soon as you say arsenic trioxide, everything's poison. Please remember that every drug is a poison. Okay, the only difference between a poison and a drug is the right dose. So when you use arsenic trioxide in the doses that we are using, we have enough data that the long-term side effects are not that significant, but there is some amount of neuropathy, some amount of decrease in counts, and the occasional patient can develop hepatotoxicity and cardiotoxicity, but those are few and far apart. It's an extremely well-tolerated drug at the doses that we use.
Okay, so I think we can take one final question. So what should be the role of medical colleges, medical schools in India to really encourage students to take up research as a career?
The honest and hard answer is complete shutdown and reboot. Okay, so I hope there was a Control Alt Delete in the entire system, but doesn't exist because we really need to. I mean, I think, you know, at least the major academic centers can come together and really try and encourage research. See, what has happened is we've become silos of medical colleges and silos of basic science research, and they don't talk to each other. And I think it's so important for medical schools either to be either basic science institutes should be integrated into medical schools, or medical schools should be integrated as part of things, and so that there's a smooth crosstalk. If you go to any US center in the world, I mean, the United States or many Western centers, you will find that they are so closely integrated with each other. You know, your next room person will be a pure basic scientist. One floor above will be, you know, another unit. You know, but here we are completely in two different silos. If we could break that, I think we'll make a lot of progress.
Thank you, Vikram, for joining us today. Before I thank you again, I would like to just add that I am not Yukti, I am Banya. But we are from the same team. So Yukti had been communicating with you. So
I'm so sorry. I've been
That's all right.
With Yukti. Okay.
Yes, we are a team of 4 at India Alliance, and we work together. So thank you again for that wonderful session and for sharing your journey with us and making some very valid points that what is important in research and how to balance a professional career with personal life. You really motivated us and inspired us today. Thank you very much for joining us.
Thank you.
And
Bye.
before we sign off,
log off?
Yes.
Okay.
Thank you. Before we sign off, I'll just make one last announcement. You all will get the e-certificates if you have been a part of the workshop today and have attended more than 90% of the workshop. But it will take us like 5 to 6 days to get back to you with the certificates. And also you will have to take the in order to get the e-certificates. These surveys will be very, very helpful for us to understand what more you would want to see in these workshops and how we can improve in these workshops. With that, I sign off, and please take care, stay safe, and take good care of yourself. Thank you.